Welcome to Dreamy Facts. As the day gently recedes, we invite you to settle into a space of calm contemplation. Tonight, we embark on a journey through the intricate world of human immune cells, exploring approximately 100 fascinating facts that reveal the subtle wisdom, profound complexity, and ceaseless dedication within our own bodies. There is no pressure to remember or retain these details. Instead, let them unfold naturally, inviting your mind to settle into a state of peaceful curiosity. This exploration is designed to be a soothing experience, a gentle current of knowledge flowing without demanding effort or attention. We will observe the intricate dance of immune cells, their subtle adaptations, and the quiet wisdom embedded in their collective function, which tirelessly guards our health. Each fact is a small window into the vast and interconnected tapestry of the human immune system, presented in a calm and meditative tone, allowing your thoughts to drift and find stillness. Allow the gentle rhythm of these discoveries to guide you towards a state of relaxation, inviting a sense of wonder and peace as you prepare for rest. If you're finding peace in this journey, a like and subscribe helps us continue creating these restful explorations, and we're always curious. Share in the comments what city and time zone you're watching from. There's something special about knowing this experience is reaching people all over the world as they prepare for rest. As we gently begin our exploration, we turn our attention to the foundational guardians of our well-being, the innate immune system. This ancient and immediate defense mechanism operates tirelessly, providing a constant vigilance against potential threats, often without us ever being aware of its profound work. It is a world of constant motion and precise, rapid response. The human immune system is a complex network of cells, tissues, and organs that work together to protect the body from harmful invaders. Research shows that it can broadly be divided into two main branches: the innate immune system and the adaptive immune system. The innate immune system, which we focus on in this chapter, provides immediate, non-specific defense against pathogens. It is the body's first line of defense, acting rapidly within minutes to hours of an infection. Scientists understand that this system does not confer lasting immunity against specific pathogens, but rather offers a generalized protective response. Current understanding suggests that the innate immune system is evolutionarily older than the adaptive system, present in nearly all multicellular organisms. It acts as a crucial sentinel, constantly surveying the body for signs of danger, whether from external microbes or internal cellular abnormalities. The effectiveness of the innate immune system relies on its ability to recognize common molecular patterns found on pathogens, rather than specific antigens. The skin, the largest organ of the human body, serves as a primary physical barrier of the innate immune system. Studies indicate that an intact skin acts as an impenetrable fortress against most microbes, preventing their entry into deeper tissues. Beyond its physical structure, the skin also possesses chemical defenses. Research shows that sweat and sebaceous glands secrete substances like lactic acid and fatty acids, which create an acidic environment on the skin's surface, inhibiting bacterial growth. Additionally, the skin produces antimicrobial peptides, such as defensins and cathelicidins, which directly kill bacteria and fungi. The constant shedding of dead skin cells also mechanically removes attached pathogens, preventing their colonization. This multi-layered defense makes the skin an incredibly effective and dynamic barrier, constantly renewing itself and actively fighting off microbial threats, illustrating a remarkable interplay of physical and biochemical strategies. Beyond the external skin, mucous membranes line various internal body cavities that are exposed to the external environment, such as the respiratory, gastrointestinal, and genitourinary tracts. These membranes also serve as critical components of the innate immune system's physical barriers. Studies show that mucous membranes produce mucus, a thick, sticky fluid that traps inhaled particles, dust, and microorganisms, preventing them from reaching underlying cells. Cilia, tiny hair-like projections in the respiratory tract, rhythmically beat to sweep the mucus-laden particles upwards and out of the body, a process known as the mucociliary escalator. Furthermore, mucus contains various antimicrobial compounds, including lysozyme, which breaks down bacterial cell walls, and lactoferrin, which binds iron, thereby starving bacteria of an essential nutrient. This active and dynamic barrier continuously cleanses and protects our internal surfaces, representing a sophisticated internal defense system that complements the skin's external protection. Macrophages are large, specialized phagocytic cells that play a central role in innate immunity. The term macrophage literally means big eater, reflecting their primary function, engulfing and digesting cellular debris, foreign substances, microbes, and cancer cells. Scientists have observed that macrophages originate from monocytes in the blood, which migrate into tissues and differentiate. They are found in almost all tissues, where they are known by various names, such as Kupffer cells in the liver and alveolar macrophages in the lungs, each adapted to their specific environment. Beyond their phagocytic activity, macrophages are also crucial antigen-presenting cells, meaning they process ingested material and display fragments on their surface to activate the adaptive immune system. Research indicates that they secrete cytokines, signaling molecules that regulate immune responses and inflammation, making them pivotal communicators within the immune network. Their long lifespan allows them to persist in tissues, providing continuous surveillance and acting as a crucial bridge between immediate and long-term immunity. Neutrophils are the most abundant type of white blood cell, constituting about 50–70% of all leukocytes, and are key components of the innate immune system's rapid response team. Studies indicate that these cells are typically the first immune cells to arrive at the site of infection or inflammation, usually within minutes to hours. They are highly motile and can quickly extravasate from blood vessels into infected tissues. Neutrophils primarily function by phagocytosis, engulfing and digesting bacteria and other pathogens. They contain granules filled with potent antimicrobial substances, such as defensins, lysozyme, and myeloperoxidase, which are released into phagosomes to destroy ingested microbes. Current understanding suggests that neutrophils have a relatively short lifespan, typically only a few days, and are terminally differentiated cells, meaning they cannot divide. Their rapid deployment and potent antimicrobial arsenal make them essential for controlling acute bacterial infections, often sacrificing themselves in the process, forming the main component of pus found at infection sites. Natural killer NK cells, guardians against intracellular threats. Natural killer NK cells are a type of lymphocyte that belongs to the innate immune system, distinct from T and B cells of the adaptive system. Research shows that NK cells are specialized in recognizing and killing cells infected with viruses, as well as some tumor cells. Unlike cytotoxic T cells, NK cells do not require prior activation or antigen presentation to recognize their targets. They possess an inherent ability to detect abnormal cells. Scientists have observed that NK cells identify infected or cancerous cells by detecting a lack of MHC class Y molecules on their surface, which healthy cells typically display. They also respond to activating signals from stress molecules on target cells. Upon recognition, NK cells release cytotoxic granules containing perforin and granzymes, which induce apoptosis, or programmed cell death, in the target cell. This mechanism prevents the replication and spread of viruses and helps in the early control of malignancy, making NK cells a vital component of the body's immediate defense against intracellular pathogens and nascent cancers. Dendritic cells, DCs, are crucial antigen-presenting cells that act as a vital bridge between the innate and adaptive immune systems. They are widely distributed throughout the body, particularly in tissues that are common sites of pathogen entry, such as the skin, where they are called Langerhans cells, mucous membranes, and lymph nodes. Research indicates that in their immature state, dendritic cells are highly efficient at capturing antigens through phagocytosis and receptor-mediated endocytosis. Upon encountering pathogens or inflammatory signals, they mature and migrate to secondary lymphoid organs, like lymph nodes, where they present processed antigens to T cells. This presentation is critical for initiating an adaptive immune response, teaching T cells to recognize and target specific pathogens. Scientists have observed that dendritic cells are the most potent antigen-presenting cells, capable of activating naive T cells, thereby orchestrating the transition from immediate non-specific defense to highly targeted, long-lasting immunity. They are thus both sentinels detecting danger and messengers, alerting the adaptive forces. Inflammation, a coordinated response to injury. Inflammation is a fundamental protective response of the innate immune system to harmful stimuli, such as pathogens, damaged cells, or irritants. It is characterized by 5 cardinal signs: heat, redness, swelling, pain, and loss of function. Research shows that the inflammatory response is initiated by immune cells, particularly mast cells and macrophages, which release chemical mediators like histamine, prostaglandins, and cytokines upon detecting injury or infection. These mediators cause local vasodilation, leading to increased blood flow, redness and heat, and increased vascular permeability, allowing fluid and immune cells to leak into the tissue, swelling. This influx of fluid, rich in clotting factors and antibodies, helps to contain the infection and initiate repair. Studies indicate that immune cells like neutrophils and macrophages are recruited to the site of inflammation, where they phagocytose pathogens and cellular debris. While essential for healing and defense, chronic or uncontrolled inflammation can be detrimental, contributing to various diseases, highlighting the delicate balance required for an effective immune response. As we conclude this initial exploration, we can appreciate the foundational strength and immediate responsiveness of the innate immune system. From the physical barriers of skin and mucous membranes to the cellular warriors like macrophages, neutrophils, NK cells, and dendritic cells, each component plays a precise role in an orchestrated defense. This intricate network of rapid-acting cells and processes offers a constant, vigilant protection, setting the stage for the more specialized defenses we will uncover in our continuing journey. The quiet dedication of these cells allows us to navigate a world teeming with potential threats, ensuring our body's internal harmony. As the gentle current of knowledge continues to flow, inviting us deeper into the intricate world of human immune cells, we find ourselves further enveloped in a space of calm contemplation. The day gently recedes, and with it, any lingering distractions, allowing our minds to settle into the quiet wonder of the body's internal workings. In the previous chapter, we marveled at the foundational strength and immediate responsiveness of the innate immune system, discovering its vigilant cellular guardians and robust physical barriers. Now, we shall explore the deeper layers of this ancient defense, uncovering the sophisticated chemical signals and specialized cells that orchestrate its silent symphony, ensuring our continuous protection. The innate immune system, in its profound wisdom, is far more than just a collection of cells and physical walls. It is a dynamic, interconnected network that deploys an array of molecular instruments and specialized cellular players to maintain our internal harmony. We will now journey into these unseen forces, appreciating the subtle yet powerful mechanisms that tirelessly guard against threats, even as we drift into a state of serene understanding. Beyond the watchful eyes of macrophages and the rapid charge of neutrophils, the innate immune system wields a potent chemical arsenal. Soluble proteins and signaling molecules that circulate throughout our body, acting as silent alarms, direct combatants, and crucial communicators. These non-cellular components are essential for amplifying responses, marking invaders for destruction, and coordinating the broader immune effort. Scientists understand that the complement system comprises over 30 soluble proteins, primarily synthesized in the liver, which circulate in the blood and tissue fluids in an inactive state. Upon activation, these proteins initiate a proteolytic cascade, much like a domino effect, leading to a potent immune response. Research shows that the complement system can be activated by three main pathways. The classical pathway, triggered by antibody-antigen complexes or directly by pathogens. The lectin pathway, activated by microbial carbohydrates, and the alternative pathway, spontaneously activated on pathogen surfaces. All three converge to amplify the immune response. Current understanding suggests that one of the critical functions of complement proteins, particularly C3b, is opsonization. This process involves coating microbial surfaces, making them more palatable and easily recognized by phagocytic cells like macrophages and neutrophils, thereby significantly enhancing their engulfment and destruction. Studies indicate that a powerful outcome of complement activation is the formation of the membrane attack complex, MAC. This complex, composed of several complement proteins, inserts itself into the pathogen's cell membrane, creating pores that disrupt osmotic balance and lead to direct lysis, effectively bursting the microbial cell. Scientists understand that certain complement fragments such as C3a and C5a are potent anaphylatoxins. These molecules act as chemoattractants for immune cells and trigger the release of histamine from mast cells and basophils, thereby increasing vascular permeability and contributing significantly to the inflammatory response, which helps recruit more immune cells to the site of infection. In the complex tapestry of immune defense, communication is paramount. Immune cells do not operate in isolation. They constantly exchange information to coordinate their actions. This vital dialogue is facilitated by a diverse family of small proteins known as cytokines and chemokines, acting as the immune system's intricate language. Research shows that cytokines are a broad and loose category of small proteins that are crucial for cell-to-cell communication. They act as molecular messengers, influencing the growth, differentiation, and activity of immune cells, orchestrating both innate and adaptive immune responses. Scientists understand that interferons, IFNs, are a specific class of cytokines, notably type 1 interferons, IFNA and IFNV, which play a critical role in antiviral defense. They are produced by infected cells and signal to neighboring cells to heighten their antiviral defenses, inhibiting viral replication and stimulating NK cell activity. Current understanding suggests that interleukins, ILs, constitute a large group of cytokines, initially thought to communicate only between leukocytes, white blood cells. They are now known to have diverse functions, including promoting the proliferation of T cells, activating B cells, and modulating inflammatory responses, showcasing their central role in both innate and adaptive immunity. Studies indicate that tumor necrosis factor alpha, TNFA, is a pro-inflammatory cytokine primarily produced by macrophages. It plays a pivotal role in systemic inflammation, stimulating acute phase reactions, inducing fever, and promoting apoptosis, programmed cell death, in some tumor cells, though excessive TNFA can contribute to pathological inflammation. Scientists understand that chemokines are a specialized subset of cytokines that act as chemoattractants. They create chemical gradients that guide the directed migration of immune cells, such as neutrophils and monocytes, from the bloodstream into infected or inflamed tissues, ensuring that the right cells arrive at the right place at the right time. Even at the molecular level, our body deploys direct combatants. Antimicrobial peptides, AMPs, are ancient components of the innate immune system, offering immediate and broad-spectrum protection. Research shows that AMPs are small, positively charged peptides produced by various cells, including epithelial cells of the skin and mucous membranes, as well as phagocytes. They exert direct antimicrobial activity by disrupting microbial cell membranes, inhibiting cell wall synthesis, or interfering with intracellular processes, making them effective against bacteria, fungi, and even some viruses. Current understanding suggests that defensins are a major family of AMPs found throughout the body. They are particularly abundant in neutrophils and the epithelial linings of the respiratory, gastrointestinal, and genitourinary tracts, forming a crucial chemical barrier against invading pathogens. While macrophages, neutrophils, NK cells, and dendritic cells are prominent players, the innate immune system also relies on other specialized cells, each contributing a unique function to the body's comprehensive defense. These cells often act as rapid sensors, mediators of inflammation, or specialized fighters against particular threats. Scientists understand that mast cells are critical immune cells residing in connective tissues, particularly near blood vessels, nerves, and beneath epithelial surfaces like the skin and mucous membranes. They are packed with granules containing histamine and other inflammatory mediators, acting as rapid responders to injury and pathogens. Research shows that upon activation, often by allergens or pathogen-associated molecular patterns, PAMPs, mast cells rapidly degranulate, releasing histamine. This potent mediator causes local vasodilation, widening of blood vessels, and increases vascular permeability, leading to the classic signs of inflammation: redness, swelling, and warmth, which facilitates the recruitment of other immune cells. Current understanding suggests that basophils are the least common type of granulocyte circulating in the bloodstream. While less abundant than mast cells, they share many similarities, including the presence of histamine-containing granules and receptors for IgE antibodies, making them key contributors to allergic reactions and antiparasitic immunity. Studies indicate that in addition to histamine, basophils also release heparin, an anticoagulant. This release may help to prevent blood clotting at the site of infection or inflammation, ensuring that other immune cells and soluble factors can more easily reach the affected tissue. Scientists understand that eosinophils are granulocytes primarily involved in the defense against multicellular parasites, such as helminths, worms, and in the pathogenesis of allergic diseases like asthma. They are characterized by their distinctive bilobed nucleus and granules that stain bright red with eosin dye. Research shows that eosinophils release a variety of highly toxic proteins from their granules, including major basic protein, MBP, eosinophil cationic protein, ECP, eosinophil-derived neurotoxin, EDN, and eosinophil peroxidase, EPO. These proteins are effective at damaging the outer layers of parasites and can also contribute to tissue damage in allergic inflammation. Current understanding suggests that NKT cells are a unique subset of T lymphocytes that share characteristics of both NK cells and conventional T cells. They recognize lipid antigens presented by CD1D molecules rather than peptide antigens presented by MHC, and can rapidly secrete large amounts of cytokines, influencing both innate and adaptive immune responses. This positions them as another crucial bridge in the immune system's intricate network. As we reflect on the multifaceted nature of the innate immune system, it becomes clear that its foundational role extends beyond immediate defense. It actively prepares the ground for and directly communicates with the adaptive immune system, the body's more specialized and memory-driven defense. The innate system's vigilance and initial responses are indispensable for initiating and shaping the adaptive response, ensuring a comprehensive and tailored defense. Scientists understand that while macrophages can present antigens, dendritic cells, DCs, are considered the most potent professional antigen-presenting cells. Their primary function is to capture antigens in peripheral tissues, process them, and then migrate to secondary lymphoid organs, such as lymph nodes, to present these processed antigens to naive T lymphocytes. Research shows that in their immature state, DCs are highly efficient at phagocytosis and pinocytosis, continuously sampling their environment for potential threats. They reside in tissues, acting as sentinels, poised to detect pathogens or signs of tissue damage. Current understanding suggests that upon encountering pathogens or inflammatory signals, immature DCs undergo a maturation process. This involves downregulating their phagocytic capacity and upregulating the expression of MHC molecules and co-stimulatory molecules which are essential for activating T cells. Simultaneously, they gain the ability to migrate to lymph nodes. Studies indicate that once in the lymph nodes, mature dendritic cells present processed antigens displayed on their MHC class I and MHC class II molecules to specific T cells. This crucial interaction, along with co-stimulatory signals, is what activates naive T cells, initiating the adaptive immune response and leading to the proliferation and differentiation of effector T cells. Scientists understand that DCs are not merely passive presenters. They actively influence the differentiation pathway of T cells, thereby dictating the type of adaptive immune response that will be generated. Depending on the context of antigen presentation and the cytokines they secrete, DCs can steer T cells towards a cell-mediated, e.g. cytotoxic T cells, or humoral, e.g. antibody production by B cells, response, or even promote immune tolerance. Research shows that innate immune cells, including dendritic cells, macrophages, and neutrophils, express a variety of pattern recognition receptors, PRRs. These receptors recognize conserved molecular structures found on pathogens, known as pathogen-associated molecular patterns, PAMPs, and host-derived danger signals called danger-associated molecular patterns, DAMPs, initiating immediate immune responses and influencing subsequent adaptive immunity. Examples include toll-like receptors, TLRs, and nod-like receptors, NLRs. As we gently conclude this exploration of the innate immune system's deeper mechanisms, we are left with a profound appreciation for its comprehensive and intricate design. From the cascading power of the complement system to the precise communication of cytokines, and from the specialized roles of mast cells and eosinophils to the pivotal bridging function of dendritic cells, the innate defense operates as a seamless, tireless guardian. It is a system of immediate action and subtle wisdom, constantly evaluating, responding, and preparing the way for the adaptive forces yet to be fully revealed. We find comfort in this ceaseless dedication, knowing that even as we rest, this intricate tapestry of defense continues its silent symphony, protecting us with unwavering resolve. In the coming chapters, we will journey further, exploring how these initial alarms and signals give rise to the highly specific and memory-driven adaptive immune response, building upon the strong foundation we have now so calmly observed. As the intricate dance of the innate immune system unfolds within us, a foundational layer of defense we have come to appreciate for its ancient wisdom and relentless vigilance, we now turn our gaze towards an even more specialized realm. The gentle current of knowledge carries us deeper, guiding us from the immediate, broad strokes of innate immunity to the exquisite precision and profound memory of the adaptive immune system. This system, often described as the body's elite special forces, learns, remembers, and custom tailors its responses, offering a level of protection that is both astonishingly effective and deeply personal. We settle into this space of calm contemplation, allowing the day's gentle recede to deepen our focus on the microscopic marvels that sustain our well-being. The adaptive immune system is not merely an addition to the innate defenses, it is a sophisticated evolution, capable of recognizing virtually any pathogen it encounters, remembering it for years and launching a swifter, more potent attack upon subsequent encounters. This capacity for learning and memory is what grants us long-term immunity to many diseases after initial exposure or vaccination. At the very heart of the adaptive immune system's remarkable capabilities lie its principal cellular architects, the lymphocytes. These white blood cells, primarily B cells and T cells, are the masterminds of specificity, diversity, and memory. Scientists understand that these cells are not born with pre-programmed knowledge of every potential threat. Rather, they possess an incredible genetic machinery that allows them to generate a vast repertoire of unique receptors, each capable of recognizing a distinct molecular pattern or antigen. This vast diversity ensures that no matter how novel a pathogen, there will likely be a lymphocyte somewhere in the body ready to recognize it. Research shows that lymphocytes undergo a rigorous education, ensuring they can distinguish between self and non-self. This self-tolerance is paramount, preventing the immune system from attacking the body's own healthy tissues, a critical safeguard against autoimmune diseases. Our journey into the adaptive world begins with the B lymphocytes, or B cells, the primary drivers of humoral immunity. This branch of adaptive immunity is named for the being, quote 5, or body fluids, as B cells primarily defend against pathogens residing outside of cells by producing specialized proteins called antibodies. Scientists understand that B cells originate in the bone marrow, where they also undergo their crucial maturation process. During this development, each B cell rearranges its DNA to produce a unique B cell receptor, BCR, on its surface. This BCR is essentially a membrane-bound antibody, a molecular antenna exquisitely designed to bind to a specific antigen. The diversity of these BCRs is staggering, with estimates suggesting the human body can produce billions of different specificities. Upon encountering its specific antigen, often with the crucial help of helper T cells, a B cell becomes activated. This activation triggers a remarkable transformation. The B cell proliferates rapidly, creating numerous clones of itself, and then differentiates into two main types of daughter cells, plasma cells and memory B cells. Plasma cells are the body's dedicated antibody factories. They are short-lived but incredibly prolific, capable of secreting thousands of antibodies per second into the bloodstream and other bodily fluids. These antibodies, also known as immunoglobulins, Ig, are Y-shaped proteins that do not directly kill pathogens, but rather mark them for destruction and neutralize their effects. Current understanding suggests that antibodies perform several vital functions. Neutralization. Antibodies can bind to toxins or pathogen surfaces, preventing them from interacting with host cells and causing harm. For instance, they can block viruses from entering cells or neutralize bacterial toxins. Opsonization. By coating pathogens, antibodies act as eat-me signals, making them more palatable for phagocytic cells like macrophages and neutrophils, thereby enhancing their clearance. Complement activation. Antibodies can initiate the classical pathway of the complement system, leading to inflammation, opsonization, and direct lysis of pathogens through the membrane attack complex, MAC, a mechanism we explored in the previous chapter. Antibody-dependent cell-mediated cytotoxicity, ADCC. Antibodies can also link target cells, like virus-infected cells, to NK cells, triggering the NK cells to kill the marked target. There are 5 major classes of antibodies, each with distinct structures and roles. IgM. Often the first antibody produced during a primary immune response found as a pentamer, 5 Y-shaped units in the bloodstream, highly effective at activating complement Ig, the most abundant antibody in serum capable of crossing the placenta to provide passive immunity to a fetus, crucial for long-term immunity, neutralization, opsonization, and complement activation. IgA, predominantly found in mucous secretions, tears, saliva, breast milk, gastrointestinal and respiratory tracts, providing mucosal immunity and protecting external surfaces. Ig, involved in allergic reactions and defense against parasites, often found bound to mast cells and basophils. IgD, primarily found on the surface of naive B cells, functioning as an antigen receptor, helping to activate B cells. Memory B cells, in contrast to plasma cells, are long-lived and reside in lymphoid tissues, poised for a rapid response should the same pathogen reappear. They retain the specific BCR for the encountered antigen. Upon re-exposure, these memory B cells can quickly differentiate into new plasma cells and more memory cells, leading to a faster, stronger, and more sustained secondary immune response, the very essence of immunological memory. While B cells patrol the body's fluids, T lymphocytes, or T cells, are the architects of cell-mediated immunity. Their primary role is to directly engage and eliminate infected cells, cancerous cells, or cells presenting foreign antigens. T cells are discerning warriors, recognizing antigens not directly on the pathogen's surface, but rather when they are presented by other cells on specialized molecules called major histocompatibility complex, MHC, proteins. Like B cells, T cells originate in the bone marrow, but their maturation journey takes them to a unique primary lymphoid organ, the thymus. Within the thymus, T cell precursors undergo a rigorous selection process, ensuring that only T cells with functional T cell receptors, TCRs, that can recognize MHC molecules and do not react strongly against self-antigens are allowed to mature and exit into circulation. This thymic education is paramount for self-tolerance. Each T cell expresses a unique T cell receptor, TCR, on its surface, analogous to the B cell receptor. However, unlike BCRs, TCRs can only recognize antigens that have been processed and presented by MHC molecules on the surface of another cell. This requirement for antigen presentation is a fundamental difference between T and B cell activation. There are several crucial subsets of T cells, each with distinct roles in the immune response. Recognizable by the CD4 co-receptor on their surface, helper T cells are perhaps the most pivotal communicators of the adaptive immune system. They do not directly kill infected cells or produce antibodies, but rather act as critical orchestrators, coordinating and amplifying the responses of other immune cells. Research shows that helper T cells recognize antigens presented on MHC class II molecules, which are typically found on professional antigen-presenting cells, APCs, like macrophages, B cells, and most potently, dendritic cells. Upon activation, helper T cells proliferate and differentiate into various effector subsets, e.g., Th1, Th2, Th17, each secreting specific cytokines, the molecular messengers we explored in the previous chapter. These cytokines direct and enhance the activity of other immune cells. They can activate B cells to produce antibodies, especially for protein antigens. They can enhance the killing capacity of macrophages. Crucially, they provide essential help to cytotoxic T cells, enabling them to mount a robust and sustained attack. Identified by the CD8 co-receptor, Cytotoxic T cells are the body's specialized assassins. Their primary mission is to directly detect and destroy cells that are infected with intracellular pathogens, like viruses or some bacteria, or that have become cancerous. Studies indicate that CTLs recognize antigens presented on MHC class I molecules, which are found on virtually all nucleated cells in the body. This ubiquitous expression of MHC class I allows CTLs to surveil every cell for signs of internal trouble. When a CTL recognizes its specific antigen presented on an MHC class Y molecule on an infected or cancerous cell, it latches on and initiates a precise killing mechanism. It releases potent cytotoxic granules containing perforin and granzymes. Perforin creates pores in the target cell's membrane, allowing granzymes to enter and trigger programmed cell death or apoptosis. This controlled self-destruction prevents the pathogen from replicating further and spreading. A specialized subset of T cells, often expressing CD4 and another marker called CD25, regulatory T cells play a critical role in maintaining immune tolerance and preventing autoimmunity. Scientists understand that TREGs act as peacekeepers, suppressing excessive or misguided immune responses. They achieve this by producing inhibitory cytokines and through direct cell-to-cell contact, ensuring that the immune system does not overreact to harmless substances or attack the body's own healthy tissues. Their subtle wisdom is essential for preventing inflammatory damage and autoimmune diseases. Just like B cells, T cells also generate memory T cells after an initial encounter with an antigen. These long-lived cells allow for a faster and more potent secondary response upon re-exposure to the same pathogen, contributing significantly to long-term immunity. The interaction between T cells and antigens is entirely dependent on MHC molecules, which act as the crucial presentation platforms. These cell surface proteins display fragments of antigens, peptides, for T cell recognition. MHC class wide molecules are found on almost all nucleated cells in the body. They typically present peptides derived from proteins synthesized inside the cell, endogenous antigens, such as viral proteins produced during an infection or mutated proteins from cancerous cells. This presentation alerts CD8+ cytotoxic T cells to intracellular threats. MHC class II molecules are primarily found on professional antigen-presenting cells, APCs, including dendritic cells, macrophages, and B cells. They present peptides derived from proteins taken up from outside the cell, exogenous antigens, through processes like phagocytosis. This presentation is essential for activating CD4+ helper T cells, which then orchestrate the broader immune response. The incredible diversity of MHC genes within the human population is a double-edged sword. It helps us resist a wide array of pathogens, but it also makes tissue matching for transplants incredibly challenging. The adaptive immune system does not operate in isolation. Its cells are constantly circulating and interacting within a network of specialized lymphoid organs. Primary lymphoid organs are where lymphocytes are born and mature. Bone marrow, the birthplace of all immune cells, including B cells, which also mature here. Thymus, the specialized Bjester, quote 9, where T cells undergo their rigorous education and maturation. Secondary lymphoid organs are the sites where mature, naive lymphocytes encounter antigens, become activated, and proliferate. Lymph nodes. small bean-shaped organs strategically located throughout the body, filtering lymph fluid. They are crucial meeting points where T cells and B cells encounter antigens brought by dendritic cells, initiating adaptive responses. Spleen. A large organ in the upper abdomen that filters blood, removing old red blood cells and serving as a major site for immune responses against blood-borne pathogens. Mucosa-associated lymphoid tissue, MELT. Diffuse lymphoid tissues found in the linings of the respiratory, gastrointestinal, and genitourinary tracts, e.g., tonsils, Peyer's patches in the small intestine. These are specialized to defend against pathogens entering through mucosal surfaces. As we transition from the innate to the adaptive, the critical role of dendritic cells, DCs, the sentinels and messengers we first encountered, becomes even more pronounced. Scientists understand that DCs are the most potent antigen-presenting cells, serving as the vital bridge between the innate and adaptive immune systems. Upon encountering pathogens in peripheral tissues, immature DCs capture antigens, process them, and then undergo maturation. They migrate from the site of infection via lymphatic vessels to the nearest secondary lymphoid organs, such as lymph nodes. Here, they present the processed antigens on both MHC class I and MHC class II molecules to naive T cells, along with crucial co-stimulatory signals. This presentation is the primary trigger for T cell activation, proliferation, and differentiation, thereby initiating the highly specific and memory-driven adaptive immune response. Without the tireless work of dendritic cells, the adaptive system would remain largely unaware of peripheral threats. We have now begun to unravel the profound complexity and subtle wisdom of the adaptive immune system. Its capacity for specificity, diversity, memory, and self-tolerance represents an extraordinary evolutionary achievement. As the quiet of the evening deepens, we carry with us an enhanced appreciation for these specialized defenders, preparing to delve further into the intricate mechanisms that allow them to learn, remember, and tirelessly guard our internal landscape. As the gentle current of knowledge carried us through the foundational architecture of adaptive immunity in our previous exploration, we began to appreciate the profound specificity of B and T lymphocytes. We understood their roles as precision instruments, crafting tailored responses and retaining a memory of past encounters. Now, as the day gently recedes and a quiet calm settles, we delve deeper into the intricate choreography that orchestrates this precision. The precise mechanisms by which T cells become activated, how the immune system generates such an astonishing diversity of receptors, and the critical safeguards it employs to ensure this powerful system recognizes and tolerates the self. We settle into this space of calm contemplation, inviting a sense of wonder as we uncover the meticulous details of our body's internal vigilance. This journey reveals not just the power of our immune cells, but their inherent wisdom in distinguishing friend from foe, a balance essential for our well-being. The activation of a naive T cell is not a simple encounter. It is a meticulously orchestrated symphony requiring multiple signals, a safeguard against unwarranted immune responses. At the heart of this process lies the major histocompatibility complex, MHC, the very platform upon which antigens are presented. Scientists understand that MHC class I molecules primarily present endogenous antigens, meaning peptides derived from proteins synthesized within the cell itself. This pathway is crucial for monitoring the health of internal cellular machinery and detecting threats like viral infections or cancerous transformations. Research shows that virtually all nucleated cells in the body express MHC class Y. Current understanding suggests that the journey of endogenous antigens begins in the cytoplasm. Here, a multi-catalytic protein complex known as the proteasome acts as a cellular shredder, breaking down intracellular proteins, including viral proteins or abnormal self-proteins, into smaller peptide fragments. This precise degradation is a critical first step in preparing antigens for presentation. Studies indicate that once peptides are generated by the proteasome, they must be transported into the endoplasmic reticulum, ER, where MHC class I molecules are synthesized and assembled. This crucial transport is facilitated by a protein complex called the transporter associated with antigen processing, TAP. TAP acts as a selective gatekeeper, ensuring only peptides of suitable length and sequence enter the ER lumen. Research shows that within the ER, nascent MHC class as heavy chains associate with a protein called V2 microglobulin. This complex then binds to the incoming peptides transported by TAP. Once a suitable peptide is loaded into the MHC class I groove, the stable MHC peptide complex can then exit the ER and be transported to the cell surface, ready to be inspected by CD8+ cytotoxic T lymphocytes. The immune system also employs a distinct pathway for exogenous antigens, those originating from outside the cell, such as bacteria or toxins that have been phagocytosed or endocytosed. Scientists understand that MHC class II molecules are specialized for presenting these external threats, primarily to CD4+ helper T cells. Unlike MHC class Y, MHC class II expression is largely restricted to professional antigen-presenting cells, APCs, like dendritic cells, macrophages, and B cells. Current understanding suggests that the process for exogenous antigens begins with their uptake by APCs through endocytosis for soluble antigens, or phagocytosis for larger particles like bacteria. Once internalized, these pathogens or their components are contained within endosomes or phagosomes. Research shows that within these acidic endosomal compartments, internalized proteins are subjected to a battery of proteases that break them down into peptide fragments. This proteolytic cleavage generates the diverse array of peptides that will eventually be presented by MHC class II molecules. To prevent MHC class II molecules from binding to self-peptides within the ER, where they are synthesized, before encountering exogenous antigens, scientists understand that a protein called the invariant chain, II, binds to the MHC class II peptide binding groove. A portion of the invariant chain, known as CLIP, class II associated invariant chain peptide, remains bound even after partial degradation of the EI, effectively blocking the groove. Current understanding suggests that as the MHC class II CLIP complex traffics to specialized endosomal compartments, a non-classical MHC molecule called HLA-DM plays a crucial role. HLA-DM facilitates the exchange of CLIP for a higher affinity exogenous peptide, ensuring that the MHC class II molecule loads the most appropriate antigen for presentation to helper T cells. The presence of an MHC peptide complex on an APC is necessary but often not sufficient to fully activate a naive T cell. Research shows that a crucial second signal, known as co-stimulation, is required. This typically involves the interaction between co-stimulatory molecules like B7, CD80, CD86 on the APC and CD28 on the T cell. Without this second signal, the T cell may become anergic, non-responsive, or undergo apoptosis, a vital mechanism to prevent autoimmune reactions. Scientists understand that the environment in which T cell activation occurs, particularly the specific cytokines secreted by the APC and other immune cells, profoundly influences the differentiation of activated helper T cells. Different cytokine profiles can steer naive CD4 plus T cells into distinct subsets, such as Th1, Th2, Th17, or regulatory T cells, TREGs, each with specialized functions in orchestrating immune responses. The adaptive immune system's ability to recognize an almost infinite array of pathogens, even those never encountered before, stems from an extraordinary genetic mechanism that generates immense diversity in T cell receptors, TCRs, and B cell receptors, BCRs. Research shows that the staggering diversity of TCRs and BCRs is primarily generated through a unique somatic gene rearrangement process called VDJ recombination. This process occurs during lymphocyte development in the primary lymphoid organs, thymus for T cells, bone marrow for B cells, and involves the selective joining of variable V, diversity, D, for TCR beta and BCR heavy chains, and joining J gene segments. Scientists understand that VDJ recombination is catalyzed by a specialized set of enzymes known as recombination activating genes, RAG1 and RAG2. These RAG enzymes recognize specific DNA sequences flanking the V, D, and J gene segments, initiating the precise cleavage and rejoining of these segments in a virtually random fashion, creating a unique receptor gene. Current understanding suggests that further diversification is introduced at the junctions where V, D, and J segments are joined. During the repair process of the DNA breaks, enzymes like terminal deoxynucleotidal transferase, TDT, can randomly add non-templated nucleotides, N-nucleotides, to the junctions. This, quote 15, dramatically increases the potential repertoire of antigen-binding sites, allowing for an even greater range of antigen specificities. Research shows that both TCRs and BCRs are composed of multiple protein chains, e.g., alpha and beta chains for TCRs, heavy and light chains for BCRs. The random pairing of these independently recombined chains further multiplies the potential diversity of antigen receptors, adding another layer to the immune system's vast recognition capabilities. For B cells, diversity isn't just a developmental process, it's an ongoing refinement. Scientists understand that after activation by antigen and helper T cells, B cells undergo somatic hypermutation in germinal centers of secondary lymphoid organs. This process introduces point mutations into the V regions of the rearranged antibody genes, leading to subtle changes in the antibody's affinity for its antigen. Current understanding suggests that somatic hypermutation is coupled with a selection process known as affinity maturation. B cells that produce receptors with higher affinity for the antigen are preferentially selected to survive and proliferate, leading to the production of increasingly potent and specific antibodies over the course of an immune response. This continuous refinement ensures optimal protection. With such an incredibly diverse repertoire of antigen receptors, the immune system faces a monumental challenge: how to avoid attacking the body's own healthy tissues, the solution lies in rigorous education and selection processes, collectively known as self-tolerance. Research shows that the most critical checkpoints for establishing self-tolerance occur during lymphocyte development in the primary lymphoid organs, the thymus for T cells and the bone marrow for B cells. This process, known as central tolerance, aims to eliminate or inactivate potentially self-reactive lymphocytes before they can mature and enter circulation. Scientists understand that in the thymus, immature T cells undergo a crucial positive selection step. T cells must be able to weakly recognize self-MHC molecules, either class 1 or class 2, on thymic epithelial cells to survive. Those that fail to recognize self-MHC undergo apoptosis, ensuring that mature T cells are MHC restricted and capable of interacting with APCs. Current understanding suggests that after positive selection, T cells undergo negative selection. T cells that bind too strongly to self-antigens presented by self-MHC molecules on thymic APCs, such as dendritic cells and macrophages, are induced to undergo apoptosis. This crucial step eliminates the vast majority of potentially autoreactive T cells, preventing them from causing autoimmune disease. Research shows that a remarkable protein called autoimmune regulator, AIR, plays a vital role in negative selection. AIR allows thymic medullary epithelial cells to express a wide array of tissue-specific self-antigens from virtually every part of the body. This ensures that developing T cells are exposed to and tolerized against a comprehensive panel of self-proteins, even those not typically found in the thymus. Similar to T cells, B cells also undergo central tolerance mechanisms in the bone marrow. Scientists understand that immature B cells that strongly bind to self-antigens present in the bone marrow are induced to undergo apoptosis, a process known as clonal deletion. This directly removes highly self-reactive B cell clones from the repertoire. Current understanding suggests that some self-reactive immature B cells are given a second chance through a process called receptor editing. Instead of undergoing apoptosis, these B cells can reactivate their RAG genes and rearrange their light chain gene segments again, attempting to produce a new B cell receptor that is no longer self-reactive. If successful, they can continue their development. If not, they are eventually deleted. Despite the rigorous checkpoints of central tolerance, some self-reactive lymphocytes inevitably escape into the periphery. Research shows that the immune system employs additional mechanisms, collectively known as peripheral tolerance, to keep these potentially dangerous cells in check and prevent autoimmunity in peripheral tissues. Scientists understand that one key mechanism of peripheral tolerance is anergy, where T cells or B cells that encounter their specific self-antigen in the absence of co-stimulation become functionally inactivated and unresponsive. Without the crucial second signal from professional APCs, these lymphocytes cannot be fully activated and are essentially rendered harmless. Current understanding highlights the critical role of regulatory T cells, TREGs, in maintaining peripheral tolerance. These specialized T cells actively suppress the activation and proliferation of other immune cells, including self-reactive T cells. TREGs achieve this through various mechanisms, including the secretion of immunosuppressive cytokines and direct cell-to-cell contact. Research shows that prolonged or excessive activation of lymphocytes, especially in the absence of a clearing infection, can trigger a programmed cell death pathway known as activation-induced cell death, AICD. This mechanism helps to contract immune responses once the threat is cleared, and can also eliminate self-reactive lymphocytes that become activated in the periphery, preventing chronic inflammation and autoimmunity. As we gently conclude this segment of our journey, we have navigated the intricate pathways of T-cell activation, marveled at the genetic ingenuity that generates an almost limitless diversity of immune receptors, and witnessed the profound wisdom embodied in the mechanisms of self-tolerance. The immune system is not merely a collection of vigilant cells, but a dynamic, self-regulating entity, constantly learning, adapting, and protecting with a precision that inspires awe. We carry this understanding forward, allowing the gentle rhythm of these discoveries to deepen our appreciation for the subtle complexities that tirelessly guard our inner world, preparing us for further contemplation as our journey continues. As the gentle current of knowledge carries us deeper into the intricate world of human immune cells, we find ourselves at the threshold of adaptive immunity's dynamic functions. In our last journey, we explored the profound mechanisms by which these specialized cells, B and T lymphocytes, are meticulously trained to recognize the infinite variety of threats, all while rigorously safeguarding the body's own precious self. We witnessed the marvel of genetic recombination creating an arsenal of unique receptors, and the critical processes of self-tolerance that prevent internal conflict. Now, with this foundation laid, we turn our gaze to the powerful actions these cells undertake, transforming recognition into decisive defense. Settle once more into this space of calm contemplation, allowing your mind to drift through the microscopic landscapes within. We are about to observe the sophisticated strategies employed by the adaptive immune system to neutralize invaders and eradicate infected cells, a symphony of precision and power that tirelessly guards our well-being. The adaptive immune system's ability to produce highly specific antibodies is one of its most remarkable feats, a cornerstone of what scientists call humoral immunity. This arm of defense is primarily orchestrated by B lymphocytes, cells that mature the bone marrow and carry unique B-cell receptors, BCRs, on their surface, each capable of recognizing a specific antigen. Scientists understand that B cells require activation to transform into antibody-producing powerhouses. This activation can occur in two primary ways. The first, and most potent, is T-dependent activation, where a B cell recognizes its specific antigen and then receives crucial help from an activated helper T cell, CD4 plus T cell, that has also recognized the same antigen or a processed peptide of it. This cooperative interaction, often occurring in secondary lymphoid organs like lymph nodes and the spleen, is essential for a robust and long-lasting immune response. Research shows that some antigens, typically large molecules with repeating subunits like certain bacterial polysaccharides, can directly activate B cells without T cell help. This is known as T-independent activation. While providing a quicker initial response, it generally leads to less potent antibodies, lacks immunological memory, and is primarily restricted to IgM antibody production. It serves as an important immediate defense against certain pathogens. Upon activation, B cells undergo a dramatic transformation, differentiating into plasma cells. These specialized cells are essentially antibody factories, dedicating their cellular machinery to the mass production and secretion of millions of copies of their specific antibody every second. Studies indicate that plasma cells are short-lived in their primary response, but can become long-lived in the bone marrow after repeated exposure, continuously secreting protective antibodies. Antibodies, also known as immunoglobulins, Ig, possess a distinct Y-shaped structure. Research revealed that each antibody molecule consists of 4 polypeptide chains, 2 identical heavy chains and 2 identical light chains, held together by disulfide bonds. The arms of the Y, known as the Fab, fragment antigen binding regions, contain the variable domains that are unique to each antibody and bind specifically to an antigen. The stem of the Y, the Fc, fragment crystallizable region, determines the antibody's class and dictates how it interacts with other immune cells and molecules. Fact 65. 5 classes of immunoglobulins, specialized roles. Scientists have identified 5 major classes of antibodies, each with unique structural features and effector functions, categorized by their heavy chains. IgM, immunoglobulin M. The first antibody produced in a primary immune response, IgM typically exists as a pentamer, 5 Y-shaped units joined together, making it highly efficient at activating the complement system. IgG, immunoglobulin G. The most abundant antibody in serum, IgG is a monomer and is crucial for long-term immunity. It can cross the placenta, providing passive immunity to the fetus, and is highly effective at neutralizing toxins, opsonizing pathogens, and activating complement. IgA, immunoglobulin A. Primarily found in mucus secretions, tears, saliva, breast milk, gastrointestinal and respiratory tracts. IgA often exists as a dimer. It plays a vital role in mucosal immunity, preventing pathogens from adhering to epithelial surfaces. G, immunoglobulin E. Present in very low concentrations, IgE is primarily associated with allergic reactions and defense against parasitic infections. It binds to mast cells and basophils, triggering the release of inflammatory mediators. Ed, immunoglobulin D. Primarily found on the surface of naive B cells, EGD serves as a B cell receptor, playing a role in B cell activation. Its exact function in secreted form is less understood. One of the most direct ways antibodies protect the body is through neutralization. Studies indicate that antibodies can bind directly to toxins, preventing them from interacting with host cells or coat viral particles and bacterial pathogens, blocking their ability to attach to and infect cells. This effectively disarms the invader before it can cause damage. Research demonstrates that antibodies can act as molecular tags, a process called opsonization. When antibodies coat the surface of a pathogen, their Fc regions become exposed, serving as binding sites for phagocytic cells like macrophages and neutrophils. This enhances the engulfment and destruction of the arced pathogen, significantly improving the efficiency of phagocytosis. Current understanding suggests that antibodies, particularly IgM and certain subclasses of IgG, can activate the classical pathway of the complement system. When antibodies bind to an antigen on a pathogen surface, they undergo a conformational change, exposing binding sites for complement proteins. This initiates a cascade of enzymatic reactions leading to the formation of the membrane attack complex, MAC, which punctures the pathogen's membrane, causing lysis. Scientists understand that antibodies can also mediate the destruction of infected or abnormal cells through a process called antibody-dependent cell-mediated cytotoxicity, ADCC. In this mechanism, antibodies bind to antigens on the surface of target cells, e.g. virally infected cells or tumor cells. Natural killer NK cells, equipped with Fc receptors, recognize and bind to the Fc region of these antibodies. This binding triggers the NK cell to release cytotoxic granules, leading to the death of the antibody-coated target cell, while humoral immunity deploys antibodies to combat extracellular threats. Cell-mediated immunity, primarily driven by T lymphocytes, targets intracellular pathogens and abnormal cells. These cells, matured in the thymus, operate with surgical precision, ensuring that infected or dangerous cells are eliminated. The CD8 plus T cells, once activated and differentiated into cytotoxic T lymphocytes, CTLs, are the immune system's specialized assassins. Research shows that CTLs are uniquely capable of recognizing and directly killing host cells that are infected with intracellular pathogens, like viruses or some bacteria, or have become cancerous. They achieve this by recognizing specific antigen peptides presented on MHC class I molecules on the surface of target cells. Scientists understand that CTLs employ a sophisticated killing mechanism involving perforin and granzymes. Upon recognizing an infected target cell, the CTL forms a tight immunological synapse. Perforin molecules are then released, polymerizing to form pores in the target cell membrane. Through these pores, granzymes, which are serine proteases, enter the target cell and initiate a cascade of events leading to programmed cell death, or apoptosis. This method ensures the infected cell is removed without causing widespread inflammation. Current understanding suggests that CTLs can also induce apoptosis in target cells through the Fas-Fasl pathway. CTLs express a molecule called Fas ligand, FasL, on their surface. When FasL binds to its receptor, Fas, present on the target cell, it triggers an apoptotic signaling pathway within the target cell, leading to its demise. This pathway provides an alternative, equally precise method for eliminating compromised cells. While not directly killing pathogens, helper T lymphocytes, CD4+ T cells, are the indispensable orchestrators of the immune response. Research indicates that these cells, which recognize antigen presented on MHC class II molecules, play a central role in coordinating both humoral and cell-mediated immunity by releasing a diverse array of cytokines. Their influence is far-reaching, guiding the activation, proliferation, and differentiation of other immune cells. Studies show that Th1 cells are a critical subset of helper T cells, characterized by their production of cytokines like interferon gamma, EFNG, and TNF-A. They are primarily involved in promoting cell-mediated immunity against intracellular pathogens, activating macrophages to enhance their phagocytic and killing capabilities, and driving the differentiation of CD8 plus T cells into CTLs. Research indicates that Th2 cells secrete cytokines such as IL-4, IL-5, and IL-13. This subset is crucial for mounting immune responses against extracellular parasites, particularly helminths, worms, by promoting Ig production from B cells and activating eosinophils and mast cells. They are also heavily implicated in allergic reactions. Scientists understand that Th17 cells produce IL-17 and IL-22, cytokines that play a vital role in immunity against extracellular bacteria and fungi, especially at mucosal surfaces like the gut and skin. They promote neutrophil recruitment and enhance the integrity of epithelial barriers, forming a robust defense against these specific threats. A crucial subset, regulatory T cells, TREGs, characterized by the expression of the transcription factor FOXP3, are essential for maintaining immune homeostasis and preventing autoimmunity. As explored in the previous chapter, they secrete immunosuppressive cytokines like IL-10 and TGF-V, actively suppressing the activity of other immune cells and dampening excessive immune responses. They are the body's internal peacekeepers, ensuring that defense does not turn into self-destruction. One of the most extraordinary hallmarks of adaptive immunity is its capacity for memory. This ability to, quote 22, past encounters with specific pathogens allows for a much faster, stronger, and more effective response upon subsequent exposure, forming the basis of long-term protection. Scientists understand that after a primary immune response, a subset of activated B cells does not differentiate into plasma cells, but instead becomes long-lived memory B cells. These cells express high-affinity B cell receptors and circulate throughout the body, poised to rapidly reactivate and differentiate into plasma cells upon re-encountering their specific antigen. Similarly, a population of memory T cells, both CD4+ and CD8+, persists after the resolution of an infection. Research shows that these memory T cells are long-lived, quiescent, and circulate widely. They possess a lower activation threshold than naive T cells, meaning they can be rapidly activated upon re-exposure to the specific antigen, quickly proliferating and differentiating into effector cells. Current understanding highlights the dramatic difference between a primary and a secondary immune response. The secondary response is characterized by its rapid onset, days instead of weeks, higher magnitude, more antibodies and effector T cells, and often a higher affinity of antibodies due to affinity maturation. This swift and potent reaction typically clears the pathogen before it can cause significant disease, providing effective immunity. The principle of vaccination is a profound application of immunological memory. By introducing a weakened or inactivated form of a pathogen, or specific pathogen components, vaccines safely trigger a primary immune response, leading to the generation of memory B and T cells without causing illness. Upon subsequent exposure to the actual pathogen, these memory cells spring into action, providing rapid and protective immunity. As we gently bring our focus back to the present, we can appreciate the immense sophistication and tireless dedication of the adaptive immune system. From the precision of antibody neutralization to the surgical strikes of CTLs and the guiding hand of helper T cells, every action is part of an exquisitely coordinated defense, and in the silent vigilance of memory cells, we find the promise of enduring protection, a testament to the body's subtle wisdom and profound complexity. Our journey through these intricate facts continues, ever deepening our awe for the foundational guardians within. As the intricate dance of adaptive immunity unfolds within the body's quiet theatre, we begin to appreciate not just its precision and specificity, but also its profound capacity for learning. The systems we have explored, the B cells crafting their exquisite antibodies, the T cells discerning between self and non-self with such meticulous care, are not merely reacting to the present moment. They are building a library of experience, an archive of past encounters that will shape all future defenses, settle deeper into this space of calm contemplation, for we are now ready to uncover one of the most remarkable features of this internal world, the enduring echo of immunological memory, a wisdom that ensures the body is always prepared, always a step ahead. Immunological memory is the adaptive immune system's ability to, quote 23, a specific pathogen after an initial encounter and mount a faster, stronger, and more effective response upon subsequent exposures. Scientists understand that this foundational principle lies at the heart of long-term immunity and is the basis for how vaccines protect us. It transforms a slow, initial learning process into a rapid, decisive defense, often preventing disease symptoms altogether. Memory B cells, generated during the primary immune response, are long-lived lymphocytes that express high-affinity B-cell receptors, BCRs, and are poised for rapid activation upon re-encounter with their specific antigen. Research shows that these cells reside predominantly in secondary lymphoid organs, such as lymph nodes and the spleen, but also circulate throughout the body. When reactivated by their cognate antigen, often with the help of memory helper T cells, they quickly differentiate into antibody-secreting plasma cells, bypassing many of the initial activation steps required for naive B cells. This swift transformation allows for a rapid surge of highly specific antibodies. Memory T cells comprise diverse subsets, including central memory T cells, TCM, effector memory T cells, TEM, and tissue-resident memory T cells, TRM, each with specialized roles in secondary immune responses. Current understanding suggests that TCM cells primarily reside in lymphoid organs, possessing a high proliferative capacity and the ability to differentiate into various effector T-cell subsets upon restimulation. TEM cells circulate in the peripheral tissues and blood, poised to exert immediate effector functions, such as cytokine production or cytotoxic killing, at the site of infection. TRM cells are unique in that they take up permanent residence in specific non-lymphoid tissues, like the skin, lungs, or gut, providing localized, immediate protection against pathogens that attempt to re-enter at mucosal or barrier sites. This multifaceted memory ensures comprehensive protection. The primary immune response occurring upon the body's first exposure to a novel antigen is characterized by a lag phase, a relatively slow rise in antibody production, initially IgM, and a lower magnitude of response. Scientists understand that this initial phase involves the activation of naive B and T cells, their clonal expansion, and differentiation into effector cells. This process takes time, often several days to weeks, during which the pathogen may cause significant disease. While crucial for establishing memory, it is a less efficient and slower defense compared to subsequent responses. The secondary immune response triggered by re-exposure to the same antigen is marked by a minimal lag phase, a significantly faster and higher magnitude antibody production, predominantly IgG, and a more effective clearance of the pathogen. Research indicates that this heightened response is due to the immediate activation of pre-existing memory B and T cells. Memory B cells rapidly differentiate into plasma cells, secreting vast quantities of high-affinity antibodies. Memory T cells quickly proliferate and execute their effector functions. This swift and potent reaction often neutralizes the pathogen, before it can cause noticeable symptoms, illustrating the profound benefit of immunological memory. Affinity maturation and class switching, processes initiated during the primary response, are crucial for the enhanced effectiveness of immunological memory, ensuring that subsequent antibody responses are both stronger and better suited to the threat. Scientists understand that during the initial encounter, B cells undergo somatic hypermutation in germinal centers, leading to the selection and expansion of clones producing antibodies with progressively higher affinity for the antigen. Simultaneously, class switching allows B cells to produce different antibody isotypes, e.g., IgG, IgA, IgE, that are optimized for various immune functions and locations within the body. Memory B cells inherit these refined characteristics, making their rapid antibody production in a secondary response exceptionally potent and targeted. Beyond memory B cells, a subset of long-lived plasma cells also contributes to sustained immunity by migrating to the bone marrow and continuously secreting low levels of antibodies for months or even years. Current understanding suggests that these specialized plasma cells serve as a constant sentinel, providing an immediate baseline of protective antibodies in circulation. While memory B cells are crucial for rapidly ramping up antibody production upon re-exposure, these long-lived plasma cells offer an enduring low-level defense, ensuring that the body is never entirely unprotected, even against pathogens not currently circulating. Vaccination leverages the principle of immunological memory by introducing attenuated or inactivated pathogens or specific pathogen components to safely induce a primary immune response without causing disease. Research shows that by presenting the immune system with these carefully selected antigens, vaccines stimulate the generation of memory B and T cells. This pre-primes the immune system, so that if the vaccinated individual later encounters the actual virulent pathogen, their body can mount a rapid and robust secondary immune response, effectively neutralizing the threat before it can establish a foothold and cause illness. Various types of vaccines have been developed, each employing different strategies to safely present antigens and elicit robust immunological memory, showcasing the ingenuity of modern immunology. Scientists understand that these include live attenuated vaccines containing a weakened form of the pathogen, like the measles, mumps, and rubella MMR vaccine, inactivated vaccines containing killed pathogens like the inactivated polio vaccine, subunit vaccines using only specific protein components of a pathogen, such as the hepatitis B vaccine, toxoid vaccines containing inactivated bacterial toxins like the tetanus vaccine, and more recently, mRNA vaccines, which deliver genetic instructions for the body's cells to produce pathogen antigens, as seen with some COVID-19 vaccines. Each type is designed to maximize immune protection while minimizing risk. Herd immunity, also known as community immunity, occurs when a significant portion of a population becomes immune to an infectious disease, thereby providing indirect protection to those who are not immune. Current understanding highlights that when a high percentage of individuals are vaccinated or have natural immunity, the chain of transmission is broken, making it difficult for a disease to spread from person to person. This collective shield protects vulnerable individuals, such as infants, the elderly, and those with compromised immune systems, who may not be able to receive vaccines or mount an adequate immune response themselves. Immunological memory is not a static state. Its duration and robustness can vary depending on the pathogen, the type of immune response elicited, and individual factors, sometimes requiring booster vaccinations to maintain protection. Research indicates that while memory to some pathogens, like measles, can be lifelong, memory to others, like influenza or tetanus, may wane over time. This diminishing memory necessitates periodic booster shots, which act as a re-exposure event, reactivating memory cells and boosting antibody levels, thereby reinforcing the body's defenses and ensuring continued protection against evolving or re-emerging threats. The intricate interplay between the innate and adaptive immune systems remains critical even during memory responses, as innate signals often guide the optimal activation and differentiation of memory lymphocytes. Scientists understand that while memory cells are primed for rapid action, their full activation and the precise shaping of their response often still rely on signals from innate immune cells, such as dendritic cells. These sentinels detect pathogen-associated molecular patterns, PAMPs, and danger-associated molecular patterns, DAMPs, releasing cytokines and co-stimulatory molecules that provide the necessary context for memory B and T cells to mount their most effective and appropriate secondary response. The two systems, though distinct, remain profoundly interconnected. As the quiet hum of these facts settles, we are left with a profound sense of the body's foresight. Immunological memory is not merely a biological quirk, it is an elegant strategy, a testament to evolution's genius, ensuring that every battle fought leaves behind not just scars, but also wisdom. It is the body's way of learning from experience, of building a history that safeguards its future. In this intricate tapestry, memory threads together past and present, providing a continuous vigilant protection. May this understanding deepen your calm and appreciation for the tireless guardians within, as the gentle current of knowledge carries us deeper into the intricate world within. We find ourselves at the threshold of another profound mystery, the remarkable balance that defines the human immune system. We have journeyed through its foundational defenses, its adaptive prowess, and its enduring memory. Now, as the day gently recedes and a quiet calm descends, we turn our gaze to the delicate art of regulation, the subtle mechanisms that ensure this powerful system remains a guardian and not an aggressor. The immune system, with its vast arsenal and ceaseless vigilance, wields immense power. Its cells are designed to seek out and destroy, to remember and react with increasing ferocity. Yet, despite this formidable capacity, our bodies are not in a constant state of internal warfare. This profound peace is not accidental. It is the result of an exquisitely tuned system of checks and balances, a symphony of regulation that ensures the immune response is both effective and appropriately restrained. It is a testament to the body's subtle wisdom, a continuous negotiation between defense and self-preservation. Research shows that without stringent regulatory mechanisms, the immune system's powerful effector functions could inflict severe damage upon healthy host tissues. This delicate control prevents autoimmune diseases, where the body mistakenly attacks its own cells, and limits collateral damage during responses to pathogens. It is a fundamental principle that underpins immune health, ensuring that the cure does not become worse than the disease. While central and peripheral tolerance mechanisms are crucial for eliminating or inactivating self-reactive lymphocytes, Scientists understand that immune tolerance is an ongoing dynamic process. It is not a one-time event but a continuous active state, maintained by a complex interplay of cells and molecules throughout the body. This sustained tolerance is particularly vital in environments like the gut, where the immune system must coexist with a vast microbial population. Current understanding suggests that in the absence of specific danger signals or co-stimulation from antigen-presenting cells, T cells encountering an antigen may become anergic, a state of functional unresponsiveness. This mechanism ensures that immune responses are not triggered by harmless self-antigens or benign environmental substances, effectively turning a blind eye to non-threats. Among the many specialized cells of the immune system, a particular subset stands out as the primary architects of immune regulation, the regulatory T cells. These cells embody the immune system's capacity for self-control, actively suppressing immune responses and maintaining a tranquil internal environment. Scientists understand that regulatory T cells, often identified by the expression of CD4, CD25, and the transcription factor FOXP3, are a specialized subset of T lymphocytes with the primary function of maintaining immune tolerance. They act as the immune system's peacekeepers, actively suppressing the activation and proliferation of other immune cells, thereby preventing excessive or autoimmune responses. Research shows that TREGs can originate in two main ways. Natural TREGs, NTREGs, develop in the thymus during T-cell maturation, undergoing a specialized selection process. Induced TREGs, iTREGs, develop in peripheral lymphoid organs from conventional CD4+ T cells in response to specific environmental cues, often in the presence of certain cytokines like TGF-beta. This dual origin ensures a robust and adaptable population of regulatory cells. Studies indicate that the transcription factor FOXP3, forkhead box protein P3, is the critical determinant of TREG lineage and function. Its expression is essential for the development and suppressive activity of TREGs. Mutations in the FOXP3 gene in humans lead to a severe autoimmune disease known as IPEX syndrome, immune dysregulation, polyendocrinopathy, MAY, enteropathy, X-linked syndrome, highlighting its indispensable role in immune tolerance. Current understanding suggests that TREGs employ a diverse array of strategies to exert their suppressive effects. These include the secretion of immunosuppressive cytokines, direct cell-to-cell contact, metabolic disruption, and even the modulation of antigen-presenting cells. This multi-pronged approach allows TREGs to effectively dampen immune activity in various contexts. Research shows that TREGs produce potent immunosuppressive cytokines, primarily interleukin-10, IL-10, and transforming growth factor beta, TGF-beta. IL-10 acts to inhibit the production of pro-inflammatory cytokines and suppresses the antigen-presenting capacity of macrophages and dendritic cells. TGF-beta has broad inhibitory effects on lymphocyte proliferation and differentiation, further contributing to immune suppression and tissue repair. Scientists understand that in addition to cytokine secretion, some TREGs can directly induce apoptosis, programmed cell death, in activated effector T cells. This is achieved through mechanisms involving granzymes and perforin, or by upregulating Fas ligand, a FASL on their surface, which binds to Fas on activated T cells, triggering their demise. This direct elimination is another powerful way TREGs maintain immune homeostasis. Studies indicate that tregs constitutively express high levels of CD25, the high-affinity alpha chain of the IL-2 receptor. By avidly binding and consuming IL-2, a cytokine crucial for the proliferation and survival of effector T cells, tregs effectively starve these cells of a vital growth factor. This metabolic competition helps to limit the expansion of immune responses. Current understanding suggests that tregs can also directly interact with and modulate the function of antigen-presenting cells, APCs, such as dendritic cells. They can reduce the expression of co-stimulatory molecules like CD80, CD86, and MHC molecules on APCs, making them less capable of activating naive T cells. This dampening of APC function is a crucial mechanism in preventing immune activation. Beyond the specialized role of tregs, the immune system employs a broader array of mechanisms to ensure its responses are proportionate and transient, preventing chronic inflammation or self-attack. Research shows that anergy is a state where T or B lymphocytes, despite encountering their specific antigen, become functionally unresponsive due to insufficient co-stimulation. This, quost25-deld, renders the cells unable to respond effectively to subsequent antigen exposure, even if proper co-stimulation is provided. It is a critical mechanism for maintaining peripheral tolerance to self-antigens. Scientists understand that activation-induced cell death, AICD, is a mechanism by which chronically activated T cells are eliminated through apoptosis. This process helps to contract the immune response after a pathogen has been cleared, preventing excessive inflammation and the perpetuation of potentially harmful effector cells. It is a crucial SEMS,26 that restores immune homeostasis. Studies indicate that immune checkpoints are a series of molecules on immune cells that, when engaged, deliver inhibitory signals, effectively acting as TOR,27 on the immune response. Two prominent examples are PD-1, programmed death 1, and CTLA-4, cytotoxic T lymphocyte-associated protein 4. These molecules prevent excessive T cell activation and are vital for maintaining self-tolerance and preventing autoimmunity. Current understanding suggests that the PD-1 receptor, expressed on activated T cells, binds to its ligands PDL1 and PDL2, which are often expressed on tumor cells, antigen-presenting cells, and other tissues. This interaction delivers an inhibitory signal to the T cell, dampening its proliferation, cytokine production, and cytotoxic activity. This pathway is a key mechanism for maintaining peripheral tolerance and preventing autoimmunity. Research shows that CTLA-4, primarily expressed on tregs and activated T cells, binds to the co-stimulatory molecules CD80 and CD86 on antigen-presenting cells with much higher affinity than CD28, the primary co-stimulatory receptor on T cells. By outcompeting CD28 for these ligands, CTLA-4 effectively blocks the co-stimulatory signal required for full T cell activation, thus inhibiting immune responses. Despite the immune system's remarkable capacity for self-regulation, this delicate balance can sometimes be disrupted. When the intricate dance of activation and suppression falters, the consequences can be profound, leading to conditions where the immune system either overreacts, underreacts, or misdirects its formidable power. Scientists understand that autoimmune diseases occur when the immune system mistakenly recognizes components of the body's own tissues as foreign and mounts an attack against them. This breakdown in self-tolerance can be influenced by a complex interplay of genetic predisposition, environmental triggers, and the failure of regulatory mechanisms like TREGs or checkpoint pathways. Studies indicate that allergies are exaggerated immune responses to otherwise harmless environmental substances known as allergens. These hypersensitivity reactions involve specific components of the immune system, often IgE antibodies and mast cells, leading to symptoms ranging from mild discomfort to life-threatening anaphylaxis. It represents an overreaction where the immune system perceives a non-threat as a significant danger. Current understanding suggests that immunodeficiencies occur when one or more components of the immune system are absent or not functioning correctly. This can be primary, genetic or inherited, or secondary, acquired due to infection, medication, or other factors. A weakened immune system leaves the body vulnerable to recurrent and severe infections, highlighting the critical importance of a robust and balanced immune defense. As we conclude this exploration of immune regulation, we are left with an even deeper appreciation for the profound complexity of the human body. The immune system is not merely a collection of formidable defenders, it is a finely tuned orchestra, capable of both immense power and exquisite restraint. Its ability to distinguish friend from foe, to respond with precision, and then to gracefully recede, is a constant marvel. May this understanding deepen your sense of wonder and bring a quiet calm as we continue our journey through the elegant tapestry of life. As the day gently recedes and the quiet deepens, we continue our journey into the intricate world of the human immune system. In our previous contemplations, we explored the delicate art of immune regulation, the profound mechanisms that ensure harmony within, preventing the body from turning upon itself. We understood that immune tolerance is not a passive state, but an active, ceaseless endeavour, guarded by specialized cells and intricate molecular checkpoints. Yet, even within this finely tuned system, challenges arise, moments when the balance falters, leading to conditions like autoimmunity or allergies. Tonight, we turn our attention to another profound internal challenge, one where the Quote 28 gang itself becomes a threat, and the immune system faces a battle of unparalleled complexity, the fight against cancer. Research shows that cancer is, at its heart, a disease of uncontrolled cell growth, where cells that once belonged to the body's harmonious tapestry begin to proliferate without restraint, ignoring the signals that govern life and death. These renegade cells, though originating from self, acquire new characteristics, making them foreign enough to potentially be recognized by the vigilant immune system. The concept of immune surveillance suggests that our immune system is constantly patrolling, detecting, and eliminating these nascent cancerous cells before they can establish themselves as a full-blown tumor. It is a testament to the immune system's foundational role as a guardian, not only against external invaders but also against internal aberrations. Scientists understand that this internal vigil is a continuous dynamic process. Every day, cells within our bodies can accumulate mutations, some of which might lead to cancerous transformation. The immune system acts as a sophisticated quality control mechanism, identifying and destroying many of these abnormal cells long before they become clinically detectable. This silent, tireless guarding against internal threats highlights the subtle wisdom embedded within our biological design. Immune surveillance is the theory that the immune system constantly monitors the body for and eliminates cells that have undergone malignant transformation, acting as a crucial defense against cancer development. This ongoing patrol is a testament to the immune system's proactive nature, a constant watchfulness that often prevents disease before it even fully manifests. The primary cellular components of the adaptive immune system, which we have come to know as precision warriors, play pivotal roles in anti-cancer immunity. Among them, the cytotoxic T lymphocytes, CTLs, or CD8 plus T cells, stand as the most direct and potent assassins of cancer cells. Just as they target virus-infected cells, CTLs are trained to recognize specific tumor-associated antigens presented on MHC class I molecules on the surface of cancerous cells. Upon recognition, they unleash their cytotoxic payload, inducing apoptosis, programmed cell death, in the rogue cells. Cytotoxic T lymphocytes, CD8 plus T cells, are the primary immune cells responsible for directly recognizing and killing cancer cells by identifying tumor-specific antigens presented on MHC class I molecules. Their ability to distinguish subtle molecular changes on the surface of cells gone awry is a cornerstone of anti-tumor immunity. However, the immune system's anti-cancer arsenal extends beyond CTLs. The natural killer, NK cells, those guardians against intracellular threats we encountered earlier, also play a critical role. Cancer cells, in an attempt to evade CTLs, often downregulate their MHC class expression, essentially making themselves, quote 29, to CD8 plus T cells. This is where NK cells shine. They possess an inherent ability to detect and kill cells that lack sufficient MHC class Y, thereby serving as a crucial backup system when cancer cells try to hide from adaptive immunity. Natural killer NK cells are vital in anti-cancer immunity, particularly by recognizing and eliminating cancer cells that have downregulated their MHC class D expression, a common evasion strategy against cytotoxic T lymphocytes. Their non-specific, yet potent, killing mechanism provides an immediate layer of defense against such cleverly disguised threats. The orchestration of this anti-cancer response relies heavily on the helper T cells, CD4 T cells, and dendritic cells, DCs. Helper T cells, acting as the conductors of the immune symphony, secrete cytokines that are essential for activating and sustaining strong CTL responses. They ensure that the cytotoxic warriors are fully armed and potent. Dendritic cells, the primary sentinels and messengers, are crucial for initiating these responses. They capture tumor antigens, process them, and then migrate to lymph nodes where they present these antigens to naive T cells, educating them to become tumor-specific CTLs. Helper T cells, CD4+, enhance anti-tumor immunity by secreting cytokines that support the activation, proliferation, and survival of cytotoxic T lymphocytes. They are the vital support system, ensuring a robust and sustained attack. Dendritic cells, DCs, are crucial for initiating anti-cancer immune responses by capturing tumor antigens, processing them, and presenting them to naive T cells in lymphoid organs, thereby priming tumor-specific cytotoxic T lymphocytes. They are the bridge, translating the subtle danger signals of cancer into a targeted immune assault. The role of macrophages in the tumor microenvironment is more nuanced, reflecting their versatile nature. Studies indicate that macrophages can adopt different phenotypes depending on the signals they receive. M1 polarized macrophages, often activated by pro-inflammatory signals, can be tumoricidal, engulfing cancer cells and presenting their antigens. Conversely, M2 polarized macrophages, often induced by tumor-derived signals, can promote tumor growth, angiogenesis, blood vessel formation, and immune suppression, becoming complicit in the cancer's progression. This dual nature highlights the complex interplay within the tumor microenvironment. Macrophages in the tumor microenvironment can exhibit dual roles, with M1 polarized macrophages being anti-tumorigenic, while M2 polarized macrophages often promote tumor growth, angiogenesis, and immune suppression. Their plasticity underscores the intricate battleground that is the tumor's immediate surroundings. Despite the immune system's remarkable capacity for surveillance and elimination, cancer cells are masters of adaptation and evasion. They evolve sophisticated strategies to escape immune detection and destruction, a process known as tumor immune evasion. One common tactic is the downregulation or loss of MHC class I molecules on their surface, making them invisible to CD8+ T cells. Other strategies involve actively suppressing the immune response. Cancer cells can secrete immunosuppressive cytokines like TGF-beta and IL-10, which inhibit the activity of immune cells and promote the differentiation of regulatory T cells, TREGs, and myeloid-derived suppressor cells, MDSCs. These cells then create an immunosuppressive shield around the tumor. Cancer cells employ various mechanisms of immune evasion, including downregulating MHC class I expression to avoid cytotoxic T lymphocytes and secreting immunosuppressive cytokines like TGF-beta and IL-10. These strategies represent a clever counter-adaptation by the rogue cells. Perhaps one of the most significant discoveries in cancer immunology is the role of immune checkpoints. These are molecules on immune cells that act as brakes, regulating the intensity and duration of immune responses to prevent autoimmunity. Cancer cells exploit these checkpoints by expressing ligands, e.g., PD-L1, that bind to receptors on T cells, e.g., PD-1, effectively switching off the T cells and preventing them from attacking the tumor. Cancer cells can evade immune attack by expressing ligands, e.g., PD-L1, that bind to immune checkpoint receptors, e.g., PD-1 or CTLA-4 on T cells, thereby deactivating the T cells and creating an immunosuppressive environment. This clever manipulation of natural regulatory pathways allows tumors to flourish under the very nose of the immune system. The tumor microenvironment, TME, is a complex ecosystem surrounding the tumor, comprising cancer cells, immune cells, macrophages, TREGs, MDSCs, fibroblasts, blood vessels, and extracellular matrix. This environment is often highly immunosuppressive, shaped by the cancer cells themselves to promote their survival and growth. Understanding the TME is crucial, as it dictates the effectiveness of immune responses and therapeutic interventions. The tumor microenvironment is a complex and often immunosuppressive ecosystem, consisting of cancer cells, immune cells, stromal cells, and blood vessels, which can hinder effective anti-tumor immune responses. It is a battleground where the rules are often set by the tumor itself. The profound insights into the immune system's interaction with cancer have revolutionized cancer treatment, giving rise to immunotherapy. This approach harnesses the body's own immune system to fight cancer, rather than directly attacking the cancer cells with chemotherapy or radiation. One of the most groundbreaking immunotherapies is immune checkpoint blockade, where drugs are used to block the brakes on T cells, like PD-1 or CTLA-4, thereby unleashing the immune system to attack the tumor. Immunotherapy is a revolutionary cancer treatment approach that leverages and enhances the body's own immune system to recognize and eliminate cancer cells. It marks a paradigm shift in oncology, viewing the immune system as a powerful ally. Immune checkpoint blockade therapies, such as those targeting PD-1, PD-L1, or CTLA-4, work by releasing the natural Has-33 on T cells, allowing them to remain active and effectively target cancer cells. These therapies have transformed the prognosis for many cancers, reactivating dormant anti-tumor immunity. Another pioneering immunotherapy is CAR T-cell therapy, where a patient's own T cells are genetically engineered in the lab to express a chimeric antigen receptor, CAR, that specifically recognizes and binds to antigens on cancer cells. These super T cells are then infused back into the patient, where they can seek out and destroy cancer cells with remarkable precision. CAR T-cell therapy involves genetically engineering a patient's own T cells with a chimeric antigen receptor, CAR, that enables them to specifically recognize and kill cancer cells, offering a highly personalized and potent treatment. This sophisticated cellular engineering represents the cutting edge of precision medicine. Beyond these, other immunotherapeutic strategies are continually being developed and refined. Therapeutic cancer vaccines aim to stimulate the immune system to generate a robust and long-lasting response against tumor-specific antigens. Oncolytic viruses are viruses engineered to selectively infect and kill cancer cells, while also stimulating an anti-tumor immune response. Therapeutic cancer vaccines aim to stimulate a patient's immune system to mount a targeted and durable immune response against specific tumor antigens, preventing recurrence or progression. These vaccines teach the immune system to remember and fight the cancer. Oncolytic viruses are engineered or naturally occurring viruses that selectively infect and lyse cancer cells, while also triggering an anti-tumor immune response in the host. They offer a dual mechanism of action: direct tumor cell killing and immune activation. The journey to understand and harness the immune system in the fight against cancer is ongoing, filled with both challenges and immense promise. Current understanding suggests that the effectiveness of these therapies can vary greatly among patients and cancer types, highlighting the personalized nature of immune responses. Researchers are tirelessly working to identify new targets, overcome resistance mechanisms, and combine different immunotherapeutic approaches to achieve even greater success. The field of cancer immunotherapy is rapidly evolving, with ongoing research focused on identifying novel immune targets, overcoming resistance mechanisms, and developing combination therapies to enhance anti-tumor efficacy. The future holds immense promise for harnessing the body's natural defenses against this complex disease. As we conclude our contemplation on the immune system's silent vigil against cancer, we are reminded once more of its profound complexity and ceaseless dedication. It is a system designed not just to defend against the external, but to maintain the integrity of the self from within. In the quiet moments before rest, let us hold a sense of wonder for this internal guardian, forever adapting, forever learning, in its intricate dance to preserve life. Tomorrow, as the gentle current of knowledge carries us forward, we will explore even more facets of this remarkable biological tapestry. For now, may your mind find peace in the intricate marvels of your own being, and may your sleep be deep and restorative.